I'm really excited to talk to you guys today about the research that I do at Case Western Reserve University. And I want to share with you specifically my vision for the future because I'm interested in understanding diabetic retinopathy so that we can design therapies for patients with diabetes so that they don't lose their vision. The disease that I'm very interested in is diabetic blindness or diabetic retinopathy. And on the left you can see normal vision, where you can see these two young kids. But on the right you can see that with retinopathy there become these large bruises on the eye, and they're those big black dots that obscure your vision. And that's the blindness that you achieve through diabetic retinopathy. I'm talking about blindness today, but some of you might not be aware of how normal vision happens. And so in normal vision, light enters the eye, and I'm showing light as the sun. Light enters the eye through a very narrow hole in your eye called the pupil. And when light enters the pupil, it passes through the lens, and it will go to the back of the eye and activate cells in the retina, which is a tissue layer in the eye. And these cells will send a signal to the optic nerve, which will travel to the brain and tell you that you're seeing something. In diabetes, the retina, which is normally functioning properly, when it's exposed to sugar, there's inflammation in the retina that's caused by diabetes, and this is characterized by an increase in pro-inflammatory proteins called caspases. This inflammation leads to the dysfunction and even the death of retinal cells, which ultimately results in the vision loss. I'm going to show you some pictures of a normal retina, and you can see here on the left, this is a retinogram, and you can have these taken at your eye doctor's appointment. And on the left, the retina is very organized, it's very pretty, it's kind of broken up into 4 pie pieces, it's just very calm, everything's happy in the retina. But when you have diabetes, and usually about 15 years of diabetes, you begin to have these small areas of bleeding in the retina. And you can see that in the middle picture. And this is when your eye doctor will tell you you have retinopathy. There's already some vision loss at this point, but what really contributes to the vision loss is the stages where there's severe bleeding into the eye that ultimately leave a massive hemorrhage and a big bruise on your retina, leading to vision loss. I'm really interested in preventing the beginning stages of this disease because there's already vision loss at this point. So we want to make sure patients aren't losing any vision loss. So that's what I focus on, is how can we prevent the beginning stages of this disease. So you might be asking, you're using this term glucose. What in the world is glucose? On the left, there's a lot of candy bars, and that's where a lot of glucose can come from. But you may not know that glucose can also come from high-starch foods like potatoes, pasta, and fried and fatty foods. And so we want to avoid those foods, especially if we're diabetics, but we want to avoid them even if we're not diabetics. Inflammation is the body's response to an injury or infection or even a toxic stimuli. But the important thing to know is that inflammation is not always a bad thing. It's the first phase of wound healing. And in this case, in this picture, you can see that there's a splinter. And when you have a splinter, your body activates inflammation to ward off infection from bacteria, but also to heal that cut in your skin. And the way that it does that is it releases specific inflammatory cytokines or chemicals like interleukin-1 beta or IL-1 beta, which is what I'll be talking about today. Caspase 1 becomes activated, and it is responsible for producing IL-1 beta, the cytokine. This IL-1 beta can bind to its receptor, which is kind of like putting a key in a lock. And then when you turn that lock, which is like downstream IL-1 signaling, we can cause cell dysfunction and/or death. And the important thing to know is chronic inflammation that lasts a very long time, equals more caspase activation and more IL-1 beta production. And if we have more caspase 1 and more IL-1 beta, we have more severe disease. And we have shown in some previous experiments that if we prevent this IL-1 beta signaling, we can prevent the progression of the disease. So this is really exciting data that we'd like to better understand. Caspases are like lawnmowers because when you cut the grass, you're keeping your lawn nice and pretty. You want it to be nice for your neighborhood. But if you cut the grass too often or too short, or if you accidentally knock over your fence or run over your mom's flowers, this is very bad. Same thing with caspases. They're often turned on to help protect us from the environment, but if we turn them on inappropriately or they stay on for too long, they can leave a path of destruction. Another way that I can show you this is through this picture that I've drawn of the working model of our research, where glucose induces caspase 1 activity. It turns on the lawnmower. The lawnmower cuts the grass or creates IL-1 beta, that inflammatory cytokine I told you about. And IL-1 beta can actually signal back to the cell in a positive feedback loop and promote a vicious cycle of caspase-1 activity. The first question we want to address in our experiments is how much caspase-1 is activated in response to high glucose over time. So we want to know how much grass is the lawnmower cutting and how many lawnmowers are turned on. We use a technique called a caspase-1 activity assay. I only want to know about the caspases that are cutting. So I take advantage of the fact that caspases cut proteins, much like grass. And we attach a fluorescent molecule to certain proteins and we feed them to the caspase that's in our cell. And when that caspase cleaves these proteins, we get light or fluorescence. And how much fluorescence that we get is how much caspase-1 activity there is. So this is the first experiment where we are trying to understand how much caspase-1 activity is activated over time in response to high glucose. So the normal glucose conditions are in the gray bars, and that's an example of a non-diabetic, or most of you hopefully that are sitting in class today have normal sugar levels. But the high glucose conditions are the green bars, and we can see that the green bars are higher than the gray bars, and that means that high glucose makes more caspase-1 activity. What we wanted to know was if we dumped IL-1 beta on the cells without high glucose, could that turn the lawnmower on or the caspase on? And you can see here in the purpley bars that if we add IL-1 beta to the cells, we can turn the lawnmower on or caspase-1 on. Minocycline prevents caspase-1 from being able to turn on. It keeps the lawnmower from being able to be turned on. So the next experiment I did was to determine if the cells were able to survive if we blocked that wave of inflammation. And you can see again, the black bar is the high glucose, and the light gray and light green and dark green bars are the drug-treated samples. And when we treated with the drugs for even just 24 hours, we were able to protect these cells from death, which is pretty exciting. So the take-home message today that I hope that I shared with you is that high glucose conditions cause inflammation, and that this inflammation is chronic and long-lasting, and it requires a positive feedback loop of IL-1 beta to induce even more inflammation. And if we can prevent this IL-1 beta production, or if we can prevent the lawnmower from being turned on or from being able to cut the grass, we might be able to design a medication that would help patients keep their vision for longer, even though they have diabetes. I did research at Case for a summer, and when I was done, I realized that this was the career for me. Being able to be creative every day in your job and ask questions that don't have answers is the most exciting thing I think I could ever be doing. I can't go to a book and find an answer. I have to figure it out for myself and test things to get answers. So I think that's why I stayed in research, and I'm really excited to continue in research and education.